Welcome to week 2 of Race Matters. This week we're going to investigate the nature of race and ancestry from a biological and genetic perspective. Organisms share many characteristics because of common ancestry. As a result, we can learn a great deal about a species if we know its evolutionary history. For example, an organism is likely to share many of its genes, metabolic pathways, and structural pathways with close relatives. Common names for organisms, such as monkey, finch, maple, oak tree, sometimes cause confusion when trying to communicate. So, to avoid this ambiguity, Linnaeus in the 1800s developed a hierarchical classification system. And of course, at the bottom of this is species, which is very specific, which gives us our scientific name, humans, Homo sapiens, for instance. But then we can move up as we get a little more general and we include more groups, and some of the more specific characteristics start to disintegrate, all the way to very vague, such as kingdom, which could be an animal kingdom or a plant kingdom, where obviously there's not a lot of relationship and characteristics, to even a greater category known as domains, which is based on genetic makeup, biochemistry, and again, those physical characteristics, which are made up of eukarya, the eukaryotes, the bacteria, and the archaea. Using this classification system makes it useful for scientists to communicate and distinguish between the millions of different species that are on the planet. This is just a way to organize so we can better understand and communicate when we're looking at different organisms. One category which you did not see in our taxonomical classification was race. What can you tell from this picture? Can you tell which race each individual belongs to? Can you tell something else about each of these individuals? Many people assume they understand the genetics of these individuals, the biochemistry of these individuals, the personality of these individuals. Now, why do we have this type of thinking? Perhaps it comes from this type of logic. What can you tell from this picture? All of these organisms have feathers. Therefore, all of these organisms lay eggs. So why can we group organisms and make predictions that work sometimes? The answer has to do with how organisms have evolved in the past. Let's take this example with these Norway maple trees. Let's say we have a population of Norway maple trees, and all of a sudden, one of those seeds, for some reason, gets blown all the way to Iceland. Not normal, very rare occurrence, but if it happens, and these two populations are isolated for a long period of time, mutations do occur during DNA replication, and eventually these accumulate. And as you can see, our leaf on the right has variegation from some of these mutations that have accumulated over time. We now have an Iceland Norway maple that has some new characteristics because its genes were isolated by a geological barrier. We now have island maples and the maples, which also on the mainland of Norway also probably accumulated some mutations and changed slightly over time. So when we were looking at the maple trees, we noticed that these two populations had been isolated and mutations occurred. That means some of these genes had slight changes in their form. Your DNA is made up of nucleotides, little chemicals, that are called adenine, thymine, guanine, and cytosine. There's different sequences. And if you change one of these, it can change the form of a gene. And that's sort of what happens with a mutation. And of course, a change in the sequence may change a physical characteristic of the organism. So could humans have the same issue? Humans left Africa about 200 to 300,000 years ago, Homo sapiens migrated out. Some into Europe, took a left turn, others took a right turn, went into Asia, as you can see, little curve there for a few of them. Some of them went over a land bridge into North America. You can see our time scale there in 200,000 years ago all the way to 12,000 years ago to 4,500 years ago. So could this give us huge changes in our genetic makeup as a species? Let's think about this. So are there differences? Well, let's look at this chimpanzee. This is one of our closest members in the primate group here. We're about 2% different when we sequence the genomes of both species, humans and chimpanzees. What's the difference in the genetic makeup of all humans? The answer is 1/10 of 1%. All humans are 99.9% genetically identical when we sequence the DNA. There's my DNA molecule again over there with those A's, T's, G's, and C's that I was rambling on about in the last slide. So the reality is we're a fairly young species. You know, organisms have been on this planet for 3.8 billion years, and you know, we're a young species. So from a genetic perspective, if we look at all humans spread out through all the planet, you know, it's like those two maple species or those two maple populations we were looking at have only been separated for a couple of years. You know, they've been separated for maybe, I don't know, 30 years. Are we going to see differences at that point between those two populations? It seems highly unlikely. So while we're a relatively young species, some of you might say, well, there is that 0.1% difference, and this is an interesting map that looks at a lot of genetic traits, and we can see that each color represents some genetic variability from other populations, and we can see each continent does have some genetic difference within that, you know, within that 0.1%. What's interesting is if we look at closer populations, we usually see a 90% difference within fairly close populations. Yet, if we were to take these continents separately, we would only see a 5% difference. So again, we see more variation within the closely related groups than within the continental groups. Now let's add something else to that. We're talking about this slight difference between our different continental populations. We see species variation is continuous at a specific geographic axis, meaning there's no discrete boundaries, right? Only continuous variation. And so if we were to have certain birds or other certain, you know, foxes or bears or whatever, we see that there's no distinct boundaries in those their phenotypes. So if we had these birds that had different feather patterns, but they're the same species and they're at the same geographic axis, you know, we're going to see continuous variation in those pattern colors. There's no discrete unit. So we now know that we have these genes spread throughout the planet. And, you know, we continue to mix and match these genes, which are not discrete, but a continuum of genes that all contribute to characteristics that, you know, do not give you a one specific phenotype, but a continuum within the population. And that's why we have this incredibly broad array of diverse looking individuals here. But genetically, really, it's the same 22,000 genes, right? Forms of the gene that we're looking at. Now, some individuals might associate race with medical conditions. Again, here's another problem with using race to come to this conclusion. Number one, if we look at the top of our graph there, these are populations that have sickle cell anemia. This is, this is common in Africa, but it's also common in India and in the Middle East. So we can't really do this by, we would classify as race. It becomes more of a regional situation. And more so, if you look at the map below, this is interesting, that shows the areas where malaria occurs. So yeah, there is a selection for this gene to give resistance to malaria, but it is not specified to one race. It's really more of a regional issue of the environmental conditions. Remember, we had a continuum of genes, so this makes perfect sense. The other areas where we don't see, that was just one form of the gene that is diagrammed in the top part of our picture. There are other forms of the gene that other populations have. So again, remember, this is a continuum when we're talking about this, but it really does bring up some interesting ethical questions. We can't use race to determine what kind of medical treatments we're going to use because race is not really controlling the genes. Other conditions are. Race tells you very little about your genetic makeup. If you were to take a genetic test, I took one last year, it gave me percentages of my ancestry from certain areas. Now, the accuracy of these tests is also somewhat weak right now because certain populations have done more genetic testing. Europe and North America, for instance, more individuals take part in these tests, and we don't have a very good genetic map for entire genome sequence in parts of Africa and parts of Asia. So when you take one of these little screenshot tests, it tells you little tidbits about your ancestry, but it's really not all that accurate. It tells you other people who've taken these types of tests. Hopefully in the future, we can have lots of individuals sequence their whole genome from each of these regions. However, it's going to tell us one thing. We're all far more alike than we are different. If you are thinking of one of these tests, also remember right now it's like a magazine article instead of having your whole genome sequence, which would be like looking at an encyclopedia with all your genetic information. But from the genomes we've compared from each of these regions, we continue to see that there is really very little genetic variation. So what can we conclude? Humans have genetic variation, but over 90% of the variation is within subgroups, not between continental groups. And remember, this 90% of variation within these subgroups, that's 90% of 0.1%. So we're talking a minuscule difference in genetic variation. Yes, geographical barriers may cause some genetic differences, but again, this is a continuum. It's not discrete units. Race does not predict major genetic differences or similarities any more than height. Race fails when it comes to biological classification. Why do we have race? It is a social construct, not a scientific construct. What can you tell from this picture? You probably can make a lot more assumptions from this picture than a blood test would give you, or the analysis of the biochemical makeup of these individuals. Race is a social construct, not a biological one. Please also watch the Howard Hughes Medical Institution video in this module on skin color distribution. It discusses the mechanism, and hopefully we can discuss the mechanism on Thursday night, as well as anything else that you'd like to talk about related to genetic variation and its impact from a biological standpoint. Thank you. Thank you for your time. And a special thank you to Dr. Summer Carroll and Dr. Taylor Newton for allowing me the opportunity to talk a little bit about the nature of race and ancestry.